A pleasant morning to all of us. We are the Group 10. Before that, please allow me to introduce my co-members. Marjorie Aguilar, Jenny B. Cabico, Julie Flor Cabico, and I am Eileen May Tabile. Our topic is all about biotechnology and genetic engineering. The biotechnology to be discussed by Miss Genevieve Cabico.
Thank you very much, Aileen, and once again, good afternoon to everyone. This time, I'll discuss biotechnology, its various forms as well as its uses and applications. So, what is biotechnology? Biotechnology uses living cells to develop or manipulate products for specific purposes, such as genetically modified foods. Biotechnology, often abbreviated as biotech, is the area of biology that uses living process organisms or systems to manufacture products or technology intended to improve the human life. So, the actual term biotechnology was introduced by Carl Eric. In 1989, Robert Budd gave an account of the fact that the father of the term biotechnology was the Hungarian agricultural engineer Carl Eric. He first coined the term biotechnology in 1919, meaning the productions of products from raw materials with the aid of living organisms. So, biotechnology is a branch of science that has advanced rapidly and has emerged as potential science for providing benefits in all fields of human welfare. It has a great impact on almost all the domains of human life, may it be health, environment, foods, or agriculture. Recent advancements have led to the multidisciplinary applicability of biotechnology. Like all technologies, biotechnology offers potentially enormous benefits, but also potential risks. Just like other emerging technologies, we cannot predict with absolutely certainty the risk of biotechnology. But the good things about this is biotechnology could help address many global problems such as climate change, an aging society, food security, energy security, infectious diseases, and many more. Next, types of biotechnology. The science of biotechnology can be broken down into subdisciplines based on common uses and applications. So here is the types of biotechnology. Red biotechnology. It involves medical processes such as getting organisms to produce new drugs and using stem cells to regenerate damaged human tissues or perhaps regrow entire organs. The green biotechnology applies to agricultural processes such as producing pest-resistant crops, disease-resistant animals, and environmentally friendly development. Next is the white biotechnology. Involves industrial processes such as the productions of new chemicals or the development of new fuels or vehicles. Yellow biotechnology refers to the process that aids food production, the most popular application being the fermentation of alcohol or cheese. Blue biotechnology encompasses processes in marine and aquatic environment such as controlling the proliferation of noxious waterborne organisms. Gray biotechnology reference the ability to use biotechnology for weapons or warfare. And the last, the gold biotechnology, also known as bioinformatics, is a cross between biological processes and computing that plays a key role in biological data. The last topic that I will be discuss is the applications of biotechnology. Here are the 9 fields of biotechnology. Number 1, biotechnology in agriculture. The application of biotechnology in agriculture has resulted in benefits to farmers, producers, and consumers. The major applicability of biotechnology in the field of agriculture and horticulture are the manufacture of biofertilizers, which prove to be more beneficial than other synthetic and chemical fertilizers. Number 2, biotechnology in food and beverage industry. Several microorganisms are used beneficially in the productions of certain foods and beverages like cheese, wine curd beer, vinegar, and etc. The underlying process behind such production is fermentation. Natural fermentation has played a vital role in human development, and it is the oldest form of production of wine. Number 3, biotechnology in environment. Environmental biotechnology promises to solve many problems related to pollution, waste disposal, and many more. Example of this is the bioremediation. Bioremediation of pollutant is an effective, simple, and more practical method of removal of Earth's pollution. Number 4, biotechnology in medicines and healthcare. Medical biotechnology is used for finding coursework genetics, getting rid of and preventing diseases. It is making huge advancement and helping millions of people. Some of the most recent uses of biological techs are work genetic, testing, drug treatments, and artificial tissue growth. Example is the vaccine and antibiotics that are very useful and helpful to humans' health. Number 5, biotechnology in energy and fuels. Nowadays, oil is the major fueling material, but it is bound to run out in forthcoming years. Also, it causes a great amount of pollution. A substitute for its being found in biofuels, which are produced from sources that are relatively clean and renewable. Example is the biofuels, an alternative environmentally friendly fuel sources. Number 6, biotechnology in productions of enzymes and biochemicals. Biofertilizers, bioherbicides, bioinsecticides, and etc. are certain biologically produced chemical compounds that involve the utilizations of microbial activities. An example of industrial biotechnology uses enzymes to make bio-based products are like chemicals, ingredients, detergents, materials, and biofuels. Number 7, other industrial applications. Industrial biotechnology is one of the most promising new approaches to pollution prevention, resource conservation, and cost reduction. It is often referred to as the third wave in biotechnology. Examples of industrial biotechnology is the biocatalyst. Biocatalysts have been developed by industrial biotechnology companies such as enzymes to synthesize chemicals. Number 8, biotechnology in forensics cases. The applications of biotechnology in forensic science involve mainly the DNA fingerprinting technique. It helps in identifications of rapists, murderers, or any other criminals based on the study of DNA. Example, in a crime scene, the outcomes from DNA profiles may be used as a part of court if the samples gathered from a crime scene may give positive results with the DNA of a suspect. This could be used as a proof that the suspect had been available at the crime scene. And last, biotechnology for conservations of nature. Biotechnology is presently used for the conservation, evaluation, and utilizations of biodiversity, particularly for important crops. For the conservation of plants species, biotechnological approaches are used. Example are the germplasm banks, seed banks, gene banks, and etc.
Next slide will be discussed by Ms. Tabale.
Thank you, Ms. Kabiko. Good day to everyone. I am here to explain about the example of biotechnology. One of the example of biotechnology is the synthetic insulin. The synthetic insulin and synthetic growth hormone and diagnostic tests is to detect various diseases are just some example of how biotechnology is impacting medicine. Biotechnology has also proved helpful in refining industrial processes, environmental cleanup, and in agricultural production. Why is insulin used to treat diabetes? Ang insulin ay nakakatulong upang makontrol ang blood glucose levels by signaling the liver and muscle and fat cells to take in glucose from the blood. Insulin therefore helps cells to take in glucose to be used for energy. If the body has sufficient energy, insulin signals the liver to take up glucose and store it as glycogen. So, Scientists make insulin by inserting a gene that codes for the insulin from protein into either yeast or bacteria. These organisms become mini biofactories and start to spit out the protein, which can then be harvested and purified. One of the new discoveries of biotechnology is that doctors can now operate on patients remotely from their computer screens, guiding robotic arms to an accuracy of a few nanometers. Genetic laboratories equipped with DNA, splicing enzymes, a mere sequence of polypeptide chains can make wonders happen. Let's move on to genetic engineering. So when we say genetic engineering, it is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Let's move to genetic manipulation. In step 1 in genetic manipulation, traditionally humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Number 1. DNA extraction. It is a method to purify DNA by using physical or chemical methods from a sample separating DNA from cell membranes, proteins, and other cellular components. Number 2. Restriction enzyme cut DNA to smaller fragments so it's easier to study or analyze. Number 3 is the making copies. It is the polymerase chain reaction, which is used to make copies of a particular gene. The fourth one is the making recombinant DNA. It is the pieces of DNA from one organisms are attached from DNA of another organism. It is step 2, which is the cell transformation. It is this transformation broadly refers to the change in phenotype of a cell due to a genetic material. It is as regards the culture cells transformation involves spontaneous or induced permanent phenotypic alterations as a result of heritable changes in DNA and consequently gene expression. The next slide is all about application of genetic engineering to be discussed by Miss Julie Flor Cabico.
Thank you for your time to introduce me, Ms. Tabule. So, there are 3 applications of genetic engineering.
The first applications of genetic engineering is medicines. This is cell make important protein products that people with disease need. Examples: insulin for diabetes, human growth hormone, and blood clotting factors. Second application of genetic engineering is genetically modified foods. Genetically modified foods have had their DNA changed using genes from other plants or animals. Also, this is produce bigger foods, feed more people, disease and pest resistance, and frost resistance. The third application of genetic engineering is cloning. Cloning is a technique scientists use to make exact genetic copies of living things. Genes, cells, tissues, and even whole animals can all be cloned. Some clones already exist in nature. Single-cell organisms like bacteria make exact copies of themselves each time they reproduce. The best example of cloning is Dolly the sheep. Dolly the sheep was a female Finnish Dorset sheep and the first mama cloned from an adult somatic cell. She was cloned by associates of the Roslin Institute in Scotland using the process of nuclear transfer from a cell taken from mammary gland.
Recombinant DNA means combining DNA or genes from different cells. Recombination occurs when 2 molecules of DNA exchange pieces of their genetic material with each other. One of the most notable examples of recombination takes place during meiosis, especially during prophase 1, when homologous chromosomes line up in pairs and swap segments of DNA. Recombinant DNA technologies, the technologies used for producing artificial DNA through the combination of different genetic materials or DNA from different sources. Recombinant DNA technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine or splice DNA from different species or to create genes with new functions.
There are 5 steps in recombinant DNA technology. First, the isolation of gene of interest or DNA sequence to gene therapy. The primary goal of recombinant DNA technology is to produce a gene or DNA sequence that carries genetic combinations or informations of value in medicine, agriculture, and in our industries. As shown at the picture, enzyme like cellulase and lysozyme is used to break down the cell wall which surrounds the cell membrane. The second step of recombinant DNA technology involves inserting the genes isolated in the first step into a suitable vector. Essentially, a vector is a carrier that can carry the gene of interest into a given cell where it's replicated as the cell divides. A vector can be described as an autonomously replicating DNA molecule, or it's capable of replicating on its own. A good example of a vector is a plasmid. This is circular DNA located in the cytoplasm or commonly known as plasmid DNA. To insert the DNA sequence or gene of interest into the vector, it's important that the ideal vector is selected and prepared. Transformation is the third step of recombinant DNA technology and involves the insertions of the recombinant DNA or modified plasmid into the host cell. The primary aim of this step is to recover large amounts of the DNA molecule. The fourth step of recombinant DNA technology aims to select host cells that contain the modified recombinant vector or modified plasmid. Some of the cells take up plasmids that underwent self-ligation while others took up plasmids with unwanted genes. Some do not take in any of the plasmids. To achieve the desired results, it's important to only select cells that contain plasmids with the gene of interest. The last step of recombinant DNA technology is aimed at increasing the productions of the desired product. Recombinant DNA technology is used to increase copies of a given gene to increase the productions of a given product. Therefore, those cells act as factories in which the product is produced.
For the next topic is reported by Ms. Aguilar.
Good day, everyone. I am Marjorie Aguilar, and I will be discussing the important aspects of genomics. Genomics is the study of structure, function, and inheritance of the genome of an organism. A major part of genomics is determining the sequence of molecules that make up the genomic deoxyribonucleic acid, or also known as DNA, the content of an organism. Ang DNA at genes ay may kinalaman sa pag-aaral ng genomics. A genome is an organism's complete set of DNA, including genes. Genes may direct the production of proteins with the assistance of enzymes and messenger molecules. These proteins make up body structures such as organs and tissues, as well as control chemical reactions and carry signals between cells. Genomics also involves the sequencing and analysis of genomes through the uses of high-throughput DNA sequencing and bioinformatics to assemble and analyze the function and structure of entire genomes.
Thus, we can now say that genomics is comparatively the new science, and in broad sense, we can describe it as an extension of genetics. Genomics provide information of genome with respect to the genome size, whole genome sequence, genome arrangement, and evolution of the genome. Genomics has three subfields, and these are the structural genomics, functional genomics, and comparative genomics. First, structural genomics. It aims to determine the structure of every protein encoded by the genome. Structural genomics represents a new direction in structural biology that is based on the goal of determining as many structures as possible, even in advance of a well-defined biological question. Second, the functional genomics. It aims to collect and use data from sequencing for describing a gene and protein functions. A functional genomics gives us an idea to understand the time and place where genes will express in different subtypes of cells, level of gene expression regulation, and interaction of genes and its product that changes in gene expression during the onset of various diseases and functional roles of different genes in cellular process. Third, the comparative genomics. Comparative genomics aims to compare genomic features between different species. Through comparing the human genome with the genomes of different organisms, researchers can better understand the structure and function of human genes and can develop new strategies in the battles against human diseases. That's why comparative genomics is very useful too. Genome sequencing projects generally use a combination of two sequencing approaches, and these are the hierarchical shotgun sequencing and whole genome shotgun sequencing. So, let us know the difference of the two. Hierarchical shotgun sequencing is a genome that is broken into larger fragments prior to sequencing, and it is also known as clone-by-clone strategy. While in whole genome shotgun sequencing, the entire genome is broken into small fragments for sequencing. And now, let us try to understand the important aspects of Human Genome Project in the field of genomics. The Human Genome Project, or also known as HGP, it is an international scientific research with the goal of determining the sequence of chemical base pairs which make up human DNA and identifying and mapping all of the genes of the human genome from both physical and functional standpoint. The Human Genome Project is very important because the project was hugely significant to biology and has influenced biological research ever since. The main tasks of the Human Genome Project were to read and record the genetic instructions contained within the human genome and provide that information to researchers worldwide freely and without restriction. The Human Genome Project also sought to develop new tools to obtain and analyze genomic data to be used beyond the project to help benefit many areas of biology. The sequence of human genome is now a crucial reference for all of human biological research. It is a template against which all human genomes are compared. Since the full human genome sequence became available to the scientific community, progress of research into human health and disease has accelerated dramatically. We now know so much more about how we function, how diseases develop, and how we can apply this knowledge to improve diagnosis and treatment. However, it has also led to many more biological questions. For for scientists to tackle, as well as social and ethical issues for us all to consider.
For the conclusion, genetic engineering, also called the genetic modification, is the direct manipulation of an organism's genome using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating or copying the genetic material of interest using molecular cloning methods or by artificially synthesizing the DNA. A construct is usually created and used to insert this DNA into the host organisms. As well as inserting genes, the process can also be used to remove or knock out genes. The new DNA can be inserted randomly or targeted to a specific part of the genome. Thank you for watching and listening, and God bless.